Topic today is secondary metabolite production by plant tissue culture technique. Plants synthesize two types of phytochemicals, primary and secondary metabolites. The primary metabolites is required for the basic function of the cell. The secondary metabolites are synthesized as a protective and defective mechanism against biotic and abiotic stress like pathogen attack, physiological conditions like drought, flood, heat stress. These secondary metabolites have a good therapeutic value like antibacterial, antimicrobial activity, anti-cancer activity. Some of them are used as nerve stimulants, natural dyes, etc. Plant tissue culture technique provide alternative source for continuous and reliable supply of these economically valuable metabolites to the pharmaceutical company, food industry, and cosmetic industry. This also prevents the depletion of plant resources. These secondary metabolites fall into different categories based on their chemical structure, like terpenoids, essential oils, sterols, lignins, etc. The figure here shows a simplified pathway of secondary metabolite synthesis in plants. The steps that are involved in secondary metabolite synthesis by plant tissue culture are: firstly, high-yielding lines of plants for secondary metabolites are screened. Explants are excised from such high-yielding varieties of plants. The explants are then surface sterilized and inoculated for callus or plant cell production. These are then subcultured to increase the biomass and then screened for the secondary metabolite production. This is then followed by initiation of suspension culture to screen the high-yielding cell lines. Here, the nutrient composition is altered by adding elicitors, plant growth hormones, changing physical parameters, etc., in order to generate exogenous stress for the plant cells, resulting in higher yield of secondary metabolite. The culture methods are then optimized for maximum secondary metabolite production, and the potential cell lines are maintained by subculturing. These cell lines are then used for scale-up in bioreactors, and the large-scale productive process is optimized. This is the simplified flowchart for secondary metabolite production. This is a list of certain bioactive components derived from plants by means of tissue culture techniques. In plants, the major building blocks for most secondary metabolites are acetyl coenzyme, shikimic acid, malonic acid, 1-deoxyzyglucose 5-phosphate, and amino acids. After the formation of the basic skeleton, further modifications result in plant species specific compounds. The production of secondary metabolite in plant cell cultures can be enhanced by treating plant cells with environmental microstress, both biotic as well as abiotic. It activates the receptors on the cell wall and cell membrane, leading to production of signaling molecules. This activates a cascade of reactions and genes involved in secondary metabolite production. The figure here depicts the effects of exogenous stress on plant cell leading to production of secondary metabolite. Production of secondary metabolites in cell cultures can be enhanced by the use of elicitors. Elicitors are compounds which in small quantities generate stress conditions in the cell line, leading to the increased production of secondary metabolites in plants. Example, sodium chloride, potassium chloride, and sorbitol abscisic acid are found to stimulate alkaloid accumulation in Catranthus roseus. Based on the nature of elicitors, they can be divided into two categories: abiotic elicitors and biotic elicitors. Abiotic elicitors are substances which are of non-biological origin and can be grouped in physical, chemical, and hormonal categories. Biotic elicitors are substances of biological origin that include polysaccharides originated from plant cell walls and microorganisms as well. Certain physical factors like light and temperature influence the production of secondary metabolites. Lights of certain wavelengths have found to enhance the production of secondary metabolites. Example, in case of anthocyanin production, wavelength of 312 nanometer and 438 nanometer have found to be effective. In case of chiconin production, darkness is preferred. Certain temperatures are also found to be favorable, like 20 to 25 degrees centigrade for chiconin production in lithospermum cultures. Certain growth regulators to enhance the secondary metabolite production. Example, 2,4-D inhibits nicotine production while kinetin promotes nicotine production. Similarly, for chiconin production too, 2,4-D inhibits its production while indole acetic acid promotes it. Here, I have enlisted certain examples wherein abiotic elicitors were used to enhance the secondary metabolite production. In case of sycamore suspension, the production of tannin catechol was enhanced by increasing the carbon to nitrogen ratio. To increase the production of chiconin dye from lithospermum culture, sucrose percentage was increased in the medium and ammonium salts were completely replaced by nitrate salts. Secondary metabolite synthesis can also be done by inducing mutation in cell lines using physical or chemical mutagens. Example, increase in production of catharathin from Catranthus roseus culture and phenolics in tobacco cell culture. Certain biotic elicitors like cell wall material extracted from fungi and fungal cells are used as elicitors to enhance the production of secondary metabolites. These are certain examples of microorganisms used as elicitors in plant cell culture to increase or enhance the secondary metabolite production. For high production of secondary metabolites from cell lines, the optimization of growth conditions is done by various methods. We would discuss about it in the following slides. The process is then designed for scale-up. The secondary metabolites synthesized are then recovered and purified by downstream processing. Different strategies are developed to maximize the production of the targeted secondary metabolite or bioactive molecule. They are: first, overexpression of the key genes that are involved in the biosynthetic pathway of the target molecule. Second, blocking the competitive branches and the degradation pathways leading to enhancing the production of the bioactive molecule. Next, inhibiting the reproductive growth of the plant, thereby increasing the vegetative growth and biomass to increase the production of targeted compounds. Biotransformation of the cells. Introduce key cells into microbes and use combinatorial biosynthesis to produce target compounds or important intermediates. Strategies to maximize the production of secondary metabolites. Overexpressing certain key genes in the metabolic pathway can lead to increased production of the secondary metabolites. Another way of increasing the production is blocking the mechanism of feedback inhibition and competitive pathways and identifying rate-limiting enzymes. The figure here is an illustration of a metabolic pathway. Here, metabolite A leads to the production of secondary metabolite of interest E through a series of intermediate metabolites B, C, D. To enhance the production of E, the steps that can be thought of is: first, increase the amount of metabolite A. It can be done by addition of precursors for production of A. Second, determine the rate determining enzymes and increase their concentration. Third, block the competitive pathways. That is, in this example, metabolite C is utilized for the production of both metabolite D and F. Blocking the competitive pathway of conversion of C to F, we can increase the amount of metabolite D, thereby enhancing the production of E. Lastly, removing any feedback inhibitions in the metabolic pathway. To improve the yield of secondary metabolites in plant cell cultures, precursor feeding is an effective approach. Precursors are compounds that exist upstream of target compound in biosynthetic pathway. Most intermediates can be used as precursors. Example, addition of phenylalanine in Salvia officinalis suspension culture stimulated the production of rosmarinic acid. However, it should be also noted that Certain precursors at high concentrations can be toxic. Isotope labeled precursors are powerful tools for elucidation of biosynthetic pathways. All secondary metabolites are derived from glucose. By identifying key enzymes and rate limiting steps, the biosynthetic pathway can be manipulated for enhanced production of secondary metabolites. When treated with specific inhibitors blocking the competitive branch, more carbon flux will be channeled to the biosynthesis of target compounds and result in improved accumulation of the target compound. The secondary metabolite production can also be enhanced by creating transgenic plants or cell lines. Example, in Sassura hair root cultures, introduction of chalcone isomerase gene increased the production of apigenin by 12-fold. Introduction of flavonoid 3,5-hydroxylase gene in rose plant with red flowers led to transgenic plants having blue-purple flowers. Another approach is by combinatorial biosynthesis. In this approach, certain genes from the secondary metabolite pathway of different organisms are introduced into a microbe. Expression of these genes occur in the microbe system, leading to the production of the target bioactive molecule. Example, genes of flavanone 3-hydroxylase and anthocyanidin synthetase had been cloned from apple species, and genes for dihydroflavanol 4-reductase and UDP glucose was cloned from anthurium. These were then introduced into E. coli. When these E. coli cultures were fed with precursor for anthocyanin biosynthesis, anthocyanin production was detected in the culture media. The figure here summarizes the various steps taken for increasing the secondary metabolite production in case of plant cell cultures. Thank you.